And we're going to hear from Ken Linthicum from the USDA Center for Medical, Agricultural, and Veterinary, talking about animal pests. So, Ken, whenever you're ready.
Yeah, veterinary entomology. So, entomology, thank you. Follow up a little bit to Matt's presentation on insects. I wanted to mention that the work I'm going to just summarize here in the next few minutes is sort of a summary of a number of people's work that we work with, and here's a list of some of those people. Just what I mentioned, we heard earlier from Keith, it's really the Earth's oceans that drive the climate, and so we spend a lot of time, believe it or not, as entomologists, studying mosquitoes, in particular, looking at the ocean, which doesn't seem like it would exactly fit. Let me just briefly touch on these three topics: climate teleconnections to vector-borne disease, our ability to forecast one mosquito transmitted disease, in particular Rift Valley fever, and then a little bit about the near future, what's been happening right now. So climate teleconnections, I'm going to briefly touch on dengue, chikungunya, Rift Valley fever, and malaria. I'm sure you've all heard of dengue and chikungunya. Am I correct? Yeah. And Rift Valley fever, maybe not. Malaria, and then, of course, Zika, which I won't talk about here, but the mosquito that transmits dengue and chikungunya is the same mosquito that transmits Zika virus. So there's tremendous building evidence about this link between ENZO events and the climate anomalies associated with that and infectious diseases, particularly those transmitted by insects, and there's a list right here. Of course, the key part of the ENZO events is the warming of the Pacific Ocean and the Indian Ocean, and those are the kind of events that trigger rainfall activities. That is, for example, depicted here in this outgoing long-wave radiation map. So the blue colors here indicate cooler than normal temperatures, indicate of convective activity. So during El Nino events, warming of Indian Ocean, tremendous rainfall in the Indian Ocean that also moves over the continent, and that has disease implications. We looked at how ENZO teleconnections actually related to rainfall events, and this is a Pearson's correlation coefficient map that actually shows that. So during ENZO events, that you can see that there's rainfall in the Pacific and also the Indian Ocean. At the same time, there's also drought effects concurrently in other places in the world, for example, in Southeast Asia and Southern Africa. So what I'm going to talk about are a couple systems, one that's dependent on temperature, or another one that's dependent on rainfall. The ability of Aedes aegypti mosquitoes, and this is actually a picture of Aedes aegypti here, and Aedes albopictus mosquitoes to transmit dengue, chikungunya, and as I mentioned, also transmit Zika and yellow fever, particularly that one species, Aedes aegypti. Also, I'm just going to briefly mention a little bit about some malaria impact based on temperature and its impact on Anopheles mosquitoes depicted here, and then Rift Valley fever, which is associated with a Culex mosquito and also an Aedes macintoshi mosquito. Going to temperature first, I just wanted to show this picture. Anybody have an idea what that's a picture of, left and right? It's a mosquito. It's a section of a mosquito. And so the head of the mosquito is here, the foregut of the mosquito is here, and the midgut's here. I depict this because when a mosquito feeds on a pathogen, it's picked up in the foregut and then it passes into the midgut where it replicates. And then if it's a suitable vector, it actually leaves that midgut. And then this is actually a dengue-infected mosquito where we stain the virus this sort of reddish color. I think it's red. Verify if I'm not correct, but anyway, the virus leaves the midgut and then infects other tissues. For example, this is thoracic ganglion, nervous tissue, but the important part is it infects salivary glands. And until the salivary glands are infected, then it cannot transmit, so it's essentially a dead-end host. But once those are infected, saliva is injected when the mosquito bites, and that's how it feeds. So that the rate of transmission, the rate of replication in the mosquito is dependent on this expression shown here, developed in different extents, but most recently probably in the late 60s. It's very dependent on temperature, so mosquitoes are dependent on the ambient temperature. The higher the temperature, the faster the virus or pathogen replicates, and the faster it gets to the salivary glands and can transmit. The significance of this for vectorial capacity is that the quicker it gets to the salivary glands, the more likely it is a mosquito will transmit. And since mosquitoes don't typically live too long, even a fairly modest increase in temperature can have a major impact on vectorial capacity. Vectorial capacity, again, represented by this expression. So looking at Aedes aegypti mosquitoes, they transmit dengue in Southeast Asia. They live in water containers that are held for drinking and for other water uses. It's a domestic mosquito almost completely in its global range. This is data from Bangkok Children's Hospital from the late 1970s to the late 1990s, and this shows dengue hemorrhagic fever cases in children reporting into Bangkok Children's Hospital, and you see various peaks during this time. Well, each of these peaks occurs just prior to a very hot, dry event with elevated temperatures, even though, of course, in Bangkok it's hot and much of the time. There are peaks in that, and even those slight peaks can cause this increased transmission of dengue, which is reflected in this kind of data. Looking now at another Aedes aegypti mosquito transmitted disease, Chikungunya virus. You probably know that in the early 2000s, an outbreak started in the island of Lamu and Mombasa off the coast of Kenya, and then spread into the Pacific Islands, eventually into South Asia, Southeast Asia, and then back into Africa again. So we looked at what environmental factors, temperature, rainfall, humidity, other things like that could impact transmission. And what we were wondering is it the same? We knew that elevated temperatures were much involved with the transmission in East Africa in the beginning, but as it moved to other continents, what was the impact? So just looking at it, in East Africa, it's an urban disease that occurs under elevated temperature conditions. But then when it went into South Asia, particularly into India, it still retained this urban and hot component, but also spread into rural areas. So it was hot involvement. In Southeast Asia, though, it became a rural and a sylvan, really a sylvan disease that really was not associated with elevated temperatures at all. This just shows the distribution of temperatures based on cases. And then when it came back to Central Africa, it became a rural disease under hot conditions. But the key here is that it was elevated temperatures that had that big impact. On the other hand, if you look at rainfall conditions, in East Africa it was under drought conditions. South Asia, drought, it could be either drought or wet conditions, anomalously dry or wet conditions. In Southeast Asia, it was the opposite. It occurred under wet conditions, over drought conditions. And in Central Africa, also a wet involvement. So here's a disease that's greatly influenced by the climate, temperature and rainfall, but it depends on what continent, what geographic location the disease occurs in, as to how that impact is. And this is interesting. It's the same virus, and it's the same mosquito, the very same mosquito, but the other components of the ecology of disease transmission has that impact. So it's a little, it's important to know that. This just shows rainfall anomalies during the outbreak time. I'll just skip through this. But it was during a drought period in 2006. This reflects it. This Hofmöller plot shows, again, chikungunya transmission during drought conditions, as opposed to this part of the graph that shows wet conditions, and in fact, that's where Rift Valley fever occurs. Just moving on here briefly to the Republic of Korea, in particular, Plasmodium vivax malaria transmission. In malaria in the Republic of Korea is depicted in this slide, and it wasn't until 1994 that malaria reappeared in the Republic of Korea, and then these lines show human cases that occurred subsequently. The bars are outgoing long-wave radiation, so indicative of, in the positive sense, hot and dry conditions. Prior to 1994, the country had been declared malaria-free. So what we see here is that starting in 1994, hot and dry conditions, the introduction of the parasite into the Anopheles mosquitoes, and then that continuation that still occurs even to the current time. And this just shows summary of continued hot and dry conditions through 2011. Depicted graphically, you can see elevated temperatures during peaks of disease transmission. Now, of course, the same thing is probably happening in the Democratic People's Republic of Korea in Northern Korea. So this is the same type of data here. And it's thought that perhaps the Plasmodium was introduced into the south from the north. As I mentioned to you, up until 1994, it was eradicated. Prior to that, though, for example, during the Korean War, there was fairly extensive malaria transmission, both in Allied troops and their opponents. And you can see here also elevated temperatures occurring here. Well, after the war, the WHO and others put extensive efforts in to try to stop malaria transmission in the Korean Peninsula. And they were quite successful. And as I mentioned, it was eradicated, declared eradicated by WHO during this period. And at this period, it's interesting to note it was also during a very cool temperature period, which certainly did help. So let me turn to a disease impacted by rainfall and not by temperature. So even though it's a virus disease that's picked up by mosquitoes, in this case it's rainfall, not temperature. In fact, temperature probably plays an inverse relationship with disease outbreaks. Rift Valley fever occurs in sub-Saharan Africa, the Middle East, the Arabian Peninsula, and it occurs during flooding events. And this shows flooding in 1997 in northeastern Kenya. It results in massive movement of people outside of those flooded areas. These people all keep animals, and they move their animals as well as themselves out of those flooded areas, mostly because they know that there's disease threat. What happens is that when these areas flood, as shown here, it brings in other animals, but what it really does is it hatches the eggs of mosquitoes that lie in the soil. Those eggs hatch, and they're already infected with the virus. And those mosquitoes come out and then start transmitting the virus. These mosquitoes collected from some of these areas, many of them will be infected. It infects cattle, sheep, and goats, causing essentially 100% abortion. So if you're in these areas, people also are greatly impacted by this. This particular family here had a mother that had lost her husband, and a daughter had lost a young baby. And they essentially lost all their animals as well. Also, people that deal with animal slaughter are at high risk. The virus is highly infectious, and coming in contact with animal tissue or blood can cause you to become infected. People that draw blood and milk for food and other reasons also are susceptible to the virus. Animal products like harvesting of skins also can contain virus and infect people. People oftentimes develop something called a hemorrhagic fever syndrome. This woman here has that. It also causes encephalitis and blindness in some cases. Animals typically abort, as I mentioned, and get sick. These are sick animals that become very lethargic, and oftentimes the adults will die as well. It has great economic impact in addition to animal health and human health. It reduces human food availability, and then of course results in high food prices and starvation. So through a number of years of work, we've been able to develop a system to actually monitor the disease, the environmental factors that influence the disease, and it relates, again, as I mentioned, to ENZO events. So these products are put on our website on a monthly basis. In 2006, we saw this ENZO impact, particularly the elevated concurrent Indian and Pacific Ocean temperatures, and similar to what we had seen in '97, and then also in the early 1980s, which we used to document the prediction model. So what we did is we put out a warning to specific areas in East Africa based on these conditions, similar to what I just showed you earlier for 1997, but this is for 2006, showing very heavy rainfall occurring in September, October of 2006 that later moved over the continent. So the warnings went out through our system to Department of Defense laboratories in Africa, to WHO, FAO, OIE, and they in turn notified regional countries. The hope was to try to mitigate what happened, and these warnings went out about 2 or 3 months before the actual outbreak was detected. FAO put together an early warning system using these products, and then ultimately there were outbreaks shown here you may have difficulty reading, but it's Kenya, Somalia, Tanzania, and then later Sudan, Madagascar, South Africa was all involved. This shows, the different dots here shows actual human cases in each of those countries that I just mentioned, and the red areas are areas that we indicated to be at high spatial risk. Some of the areas or some of the cases occurred in areas identified as non-risk, but they're all within almost walking distance of risk areas. Of course, people move to seek medical help at the closest place possible, which may be well beyond walking distance, or it could be many days walking. So spatially it was very good, but the real strength of it was the timely prediction. So in each of these graphs, you can see the warning went out at this period, and the first case was detected here. This was for Kenya. In fact, the reason this case was detected is because of the warning. A U.S. Army group in Nairobi went out to the area at risk and started looking for the virus in animals and people, and were able to find it. The same warning went out for Tanzania, which occurred a little bit later, and then and the same was true for Sudan. So from 2006 to 2011, this picture shows the area at risk, and then again the dots, the blue and yellow dots, are indicating human cases. And I have to point out, this is really an animal disease, and because of poor animal surveillance, we have to rely on human cases, and you can see there are a lot of them. But what happens with the animals, we're assuming also coincides with the people. So what happened here in 2015 into this year? Of course, we know another ENZO event. This is the long-term history of ENZO back, I think this is to 1982. And then the ocean temperatures here. In 2015, we again saw this quite elevated Pacific and Indian Ocean concurrent elevation in temperatures indicative of a potential threat. So if you compare what was happening in 2014, 15, and then into 2016 to 2006, 2007, it was very comparable. So again, back to the ENZO connections. So we put out warnings again for heavy rainfall in the Horn of Africa, and we were sure that there would be Rift Valley fever outbreak. But also during this time, we had related other diseases. For example, we thought also there would be dengue transmission in Southeast Asia because of the elevated temperatures. And in fact, this just shows some of the conditions. So this actually went out in 2015. You can see that we're actually talking about diseases occurring at many different parts of the world based on the upcoming ENZO event. Cholera and malaria in India, cholera and malaria and Rift Valley fever in Africa. Chikungunya and dengue were high on our list in this part of Brazil. Also increased malaria transmission, and then also increased chikungunya and dengue transmission in the Middle Americas, including Florida, and then hantavirus with pulmonary syndrome and West Nile virus increased transmission in the west coast of the U.S. So we did this, and we put out this this is sort of a more detailed description of the different geographic areas and what were thought to be diseases at increased risk. So this is just a map that shows some of the occurrences of what happened in 2016. As it turned out, we were quite accurate in both the spatial and the temporal predictions of this. However, to my surprise, there was no Rift Valley fever occurring in the Horn of Africa. And after spending one lifetime doing this, I was a little disappointed. Anyway, so I went there, and this is what I found. I found flooded mosquito habitats, typical of what I had seen over the last 30 or more years. And then I rolled up my pant leg, and Aedes McIntoshi mosquitoes got on me and blood fed. And so I'm thinking, well, okay, so environmentally everything's right. The mosquitoes there, the mosquitoes out. But this is what the sheep look like there. They were perfectly okay. So what happened? The climate was as expected. The ecological conditions were as expected, but this is the epidemic cycle as we hypothesize it to be, and then here's the endemic cycle. What happens is, as I mentioned to you, mosquito eggs that are lying in these areas flood during this flooding event. If it stays flooded long enough, there's two kinds of mosquitoes that will come out. The first mosquito, the Aedes mosquito, will infect some of the animals, but if it's flooded long enough, the Culex mosquito will come out and then become infected from all the other animals there, and it will cause an epidemic. So there's a couple of things you could do. Well, you can't stop the rain, but you could interfere with the hatching of the eggs in these areas. There are natural and non-natural ingredients you can put in these areas. We had hoped something like that might happen, but that didn't happen. And then there's another way that you can actually stop mosquitoes from feeding on animals by use of repellents, and the same is, of course, true for people. But what did happen was, after talking to the farm manager and then later the veterinary authorities there, they because of our warnings, they did mass vaccination on their own without telling FAO or OIE, and vaccinated millions of animals. Essentially, all animals in the country were vaccinated. And even when I was there, they were still vaccinating. So I think it's safe to say that it stopped the transmission. So vaccination occurred there. So this is a typical virus cycle, that this is numbers of cases of a vector-borne disease that would happen. Again, the key would be to have an early detection system where you could institute vector control, which could actually happen even before the rain event. Or perhaps what I thought would be even more difficult would be to do a mass vaccination campaign, but they did, and it was effective. So I just wanted to mention a couple other things here. And that is, this was the occurrence of the geographic spread of Zika virus in late 2015 into 2016. This is what the world's conditions looked like at that time. And it's similar to what we had thought before. Hot and dry conditions in Brazil existed from October to December of 2015. What role that played in Zika transmission, of course, we're yet to find out. But there was anticipated increased chikungunya transmission because of those conditions, which also spread into the Caribbean. Now you may or may not know that there was a very large dengue outbreak in the island of Hawaii this year. And the points on this map actually show where dengue was transmitted on the island of Hawaii. And that's a big event. But it got overshadowed by other events, chikungunya transmission in Florida, as well as the neotropics, and then more recently, Zika transmission. Conditions in Hawaii are shown, I think, by this circle here. Again, hot and dry conditions existed during all this transmission. And now I'm just changing subjects slightly. This is California, and something happened very unusual in California the last couple of years, but particularly this year. And Aedes albopictus was introduced into California, or I should say it was discovered in 2011 in the Central Valley of California. It actually had been introduced into a couple, well, about 15 nurseries in Los Angeles in lucky bamboo that was being imported into China in 2001. We thought we had eradicated all sources of that Aedes albopictus, But then, you know, we were looking over the Los Angeles basin and we were thinking, well, we may have missed some. But then about 10 years later, it actually popped up. So it actually became established there. Aedes aegypti had also never occurred in California that we know of. And then this is what happened in this past year. It's also, I just wanted to point out, this is the kind of conditions that were occurring in the country about the time all these infestations occurred in California. The unfortunate part is that once they become established, and by the time they're detected, they're established, it is very, very difficult to remove them. So just to summarize, our research has showed that really significant weather events can have major impact on vector-borne disease. They may have an impact also on invasive ability of certain species. But once they become established and we're dealing with them, it's much like what has been described earlier. It's a difficult process to have much impact. But if you do have the ability to detect those events before they happen, there can be very positive consequences. So thank you.
Thank you, Ken. Anyone have any question Chonggang?
Ken, it's a great presentation. I'm really interested to see the climate connections, and that can be used to do the warning system for different part of the globe. And I have been actually a postdoc at North Carolina State University with Fred Good and Alan Lloyd. Actually, trying to develop this spatially exposed mosquito population models, Aedes aegypti. And I'm just wondering, like, right now it's only based the warming is only based on the climate condition. If it's possible that when we have a more detailed information on the hydrology, the mosquito population dynamics, will be have a more detailed level of warnings for different parts of the city for something like that. Do you think that's a useful direction we can go, or that's just too complex?
Well, those are good points. I mean, my linkage to climate and invasions is very speculative at this time. But I think the link, though, to disease transmission and climate is much more solid. There are many factors. Aegypti is, of course, a very bizarre mosquito in many ways, completely domestic, except for a few remnant populations in Africa where it came from. It's adapted, and it only lives where people live, and it lives essentially in artificial containers that people develop. And it moves in those same containers. So the mosquito moves in those containers, and then of course people move with various pathogens. And then when they come together, then you have that. So yeah, there are many components, and aegypti is, I think, very challenging to try to predict. But so many things happen that we never would have guessed. The movement of chikungunya to the New World, we wondered about and couldn't believe it happened, and then of course Zika, even more surprising. So, thank you.
Anyone else? So Jeff Hickey.
Hi, thanks for your presentation. I'm curious how far out in time you are comfortable making predictions, and does that vary in space? That is it limited? Do you feel more comfortable making predictions for areas that are influenced more by El Nino, and less about areas that are less influenced by El Nino? Or
yeah, in our case, the ENSO impact seems very significant. So we feel fairly comfortable about that. But in between ENSO events, local weather, local climate can have an impact also. But it certainly is something that still needs to be looked at extensively, and we have not done so.
Did I miss anyone? Yeah, Matt.
Matt Ayers. I'm wondering about the mechanisms for precipitation and temperature effects on mosquitoes. So, for example, precipitation might favor mosquitoes by increasing habitat availability. Clearly that's true with your Rift Valley example. But it could also influence the hours of the day that are suitable for flight because of the relative humidity. And temperatures might affect the vector competence, or it might affect the development time of the immature stages, or a whole bunch of other things. What do you think are, I'm sure a lot of those things happen to some degree, but what are the mechanisms that you think are really prominent in transducing weather into changes in mosquitoes and disease?
Yeah, that's an excellent point, Matt. That equation I showed you, of course, factors in all these different components: survival, fecundity. All those are affected by climate as well. I'm not sure that I can say which is the most important. It's certainly a combination of all those factors. To try to model these different factors is certainly challenging. We attempt to do so. Clearly, in Rift Valley fever, it's a rainfall event. In the case of dengue in Southeast Asia, I think it's clearly a temperature impact. With chikungunya, it depends where it happens, but we don't understand all the reasons why. So I can't, I guess I can't say, but all those factors have to be considered, and that's probably many other factors that we have no idea that need to be discovered and then factored in.
So Ken, I had one last question. You talked about the mass vaccination of animals. Was that also done to humans? I mean, what precautions were taken with humans? Because that was part of that factor too.
The warnings went out to the population, so people were aware and tried to avoid mosquito bites, but there was no vaccination of people. In fact, there is no licensed vaccine for people. But by vaccinating the animals, you stop the source of the viremia for mosquitoes that would feed on people. So it prevented that essentially completely. If there is no amplification of the virus, then people don't get infected, unless you happen to be right at one of these locations where the mosquitoes were coming out, like I was, and where the mosquito was feeding on me. I'm okay because I've had a vaccine based on my laboratory work, experimental vaccine. But if you were there, you could possibly become infected, but most people don't get infected in that sense. They get infected after mosquitoes feed on viremic animals.
Okay, great.
Yeah.
Is the virus not harbored in non-domesticated animals? So in other words, the wild animals in the area, do they have the virus?
They get infected, but they don't become viremic, we think, at least not to high levels enough to infect mosquitoes. It's somewhat of a hypothesis. Probably the hypothesis that we have is that the domestic animals are fairly new to the ecology. Wild animals, of course, have lived with the mosquito and the disease for a long time, and probably the ones that survived by natural selection get infected, but they develop an immune response to prevent much pathology, and probably limit the viremia below what is needed to infect a mosquito.
Great, thank you. So we're going to move
Thank you.
on to our final speaker for the session today, and that is Chonggang Xu from Los Alamos National Laboratory, and he's going to talk about modeling and some of the other work that he's been doing.
Okay, great. So I really enjoyed the workshop by far, and I learned a lot. And clearly from Kate's presentation, there's a large amount of uncertainty in terms of prediction of the future climate across different models. And from Matt's presentation, we see that southern pine beetles could be very important in terms of their capability to kill the trees, even in New Mexico, not New Mexico, New Jersey. So the question will be whether we have incorporated those forest insects into the system models. And as far as I know, we know that we do not have the insect population dynamic incorporated in the current global climate models or Earth system models yet. So clearly that's not an easy job, and I think Jeff Hickey, like in the audience, he have some of the research in that area. And we recently got a grant trying to incorporate the insect population dynamic in DOE-sponsored system model, and we hired a new postdoc, Dan Johnson, and also in collaboration with Brian Alkema from University of Minnesota in the Department of Entomology. And we started this project about 1 year, and here we'd like to share our opinion on the challenges and opportunity to incorporate the insect population dynamic in the Earth system models. And at Los Alamos National Lab, we have been focused on the vegetation mortality research in the past 10 to 15 years, led by Ned McDowell. And this is a photo showing that the MEC drought back into 2002 to 3 killed millions of the pinyon pines in Los Alamos area, and that's a key motivation for Orlando or Los Alamos National Lab to be involved in this research area. And to better understand the tree physiologists and the drought conditions, we built the world's largest drought and heat experiment to help us better understand the mechanism causing the mortality. And quickly we found out that the insect play a very important role. Whether you incorporate insects or not will affect our capability to predict the tree mortality or not. And that's the reason we want to study insects. And the main goal as a DOE national lab is trying to reduce the uncertainties in the threshold carbon sink in the Earth system models. And this is a figure shown by Fred and Stan in 2014, looking at the uncertainties of the carbon fluxes from land and the carbon fluxes from the ocean. And clearly there's a large amount of uncertainties in the land carbon fluxes. And the question is, how can we reduce this amount of uncertainty? And based on very recent land surface model called the COMAD, we did sensitivity analysis. And what we found is that in terms of the carbon fluxes and the carbon states in that model, one important parameter called target carbon storage, which is the key parameter to predict the vegetation mortality under the stress, either because drought or light limitations, and clearly indicates that we want to improve the representation of the mortality in the system models. We want to improve the prediction of land carbon fluxes into the future. So we have a review paper published back in 2011 looking at what the current mortality mechanism represent in the system models. And there's different ways to represent them, but in general, they are very simple. For example, one of the popular model called community land model, they have fixed background mortality, and that's it. And some of the models be more advanced. For example, they simulate the mortality depending on growth rate, the high growth rate, the low mortality, and vice versa. But none of them, by far, have incorporated insect population dynamics. And clearly, they can ignore a lot of tree mortality resulting from the insect outbreak in this case. And that may be one of the reasons there's a large amount of uncertainty in the current system model predictions of land carbon fluxes. So we look into the data of the past, trying to see what's the importance of insects in terms of their impact on tree mortality. And luckily, we have the world's most advanced forest inventory data in US. So we mainly plot the percentage of mortality in terms of basal area for the whole USA. And there's some gaps because we do not have the recent forest inventory data. And on the western side, I think we found that the percentage of mortality of trees resulted from insects reach about more than 50%. That's a huge amount. And on the south part, on the east part, we see a less percentage, but the insect impact is universal across the whole country. And that indicates we may want to incorporate the insect-induced mortality into the Earth system model because the large amount of impact across the whole continent of the US. And if you look into the future, they may also have an increased impact on the forest mortality. And this is the figure by Bob Banks. He is here right now. So looking at the development rate for different life stages for the mountain pine beetles, and then clearly from egg to the adults. And normally the larvae and the pupae stages, they are developing in the winter and also spring. So the future temperature increase by global warming can substantially impact the development rate of insects and lead to earlier adult emergence, for example. And in extreme cases can lead to multiple life cycles and more insects on the trees. And right now we do not have evidence of the multiple life cycles for the mountain pine beetles, but we do have evidence from our collaborator, Brent Alkmaar's lab, looking at eastern large beetle life cycles. And what they found that there's multiple life cycles of the large beetle and multiple attacks in the summer. So, and that could be one reason they found that there's increasing Tamrock forest affected by the large beetles since 2000 to 2015, even though we need more evidence or more data to confirm this link. And another impact of the future climate on the insect-caused tree mortality is that the winter temperature. I think Matt pointed a lot on that side. And a good example is the mountain pine beetles. Historically, the mountain pine beetle is in this area, and we saw the mountain pine beetle have expanded to the north, but with further global warming, then the mountain pine beetle can jump from the west to the east of North America, and that could produce a huge amount of economic impact on those areas. But the climate impact may not always positive on the insect development. They potentially have some negative component. That's the heat wave, basically. So for example, mountain pine beetles, they build their anti-freeze compounds in the winter. And when the temperature became high, then they can build less anti-freezing compounds, so that the temperature leading to 50% of mortality may track the air temperature. So basically, a higher air temperature, they may build less anti-freezing compound, so that the temperature can, the half lethal temperature could be higher. And in the future, potentially, even though we got a overall mean is warmer, the variability could be much more at the same time. So potentially have more risk of the heat wave. If you have a heat wave suddenly and then there's not enough heat anti-freeze compounds, then if the temperature drops, then that can cause substantial mortality. And that could be one potential negative feedback to the insect population dynamics. And I just did one model of the 22 SSM models I've studied, and looking at the potential heat wave. This is the maximum temperature difference the difference of the previous month's maximum temperature to the current month's the minimum temperature. And that's a range compared to historic period from 1850 to 2000 based on the GFDAO model. I think Case probably knows this better. But in the US, we do see a potential of a high risk of the heat waves in the future, and that may have a negative impact on the insect population dynamics. So another key component of looking into the future is that the vegetation part, because the insect outbreak is the combination of both the insect populations and also the vulnerability of the vegetation. In the future global warming, there's more possibility of the extreme event. In this case, we're looking at the relative probability of the extreme drought event and defined by soil moisture happening only once every 50 years. And in RCP 8.5, we can see that there's about 5 times more frequent of those drought events to happen in the next 100 years. And if you look at RCP 4.5, there's about 3 times more of those extreme drought events to happen. And that could have a substantial impact on the vegetation productivity. In this case, we're looking at the net primary production, relative reduction in the future compared to the historical period. And then it's same, because the drought frequency increases, we can see that also about 5 times increase in the MPP reduction due to the drought, and about 3 times reduction for the RCP 4.5, which is the more moderate greenhouse emission scenarios. So if those drought frequency happens as predicted, then potentially this vegetation became more vulnerable to the insect outbreak, and that can substantially kill a large amount of trees. And this is just an example showing that in the Southwest, because we have the drought experiment, we built a mechanistic model of the tree vulnerability to drought, and we predict that for the pinyon pines, we saw an increase of mortality event in the next 100 years on the southern part of their current distribution. So that also potentially more pinyon apis bark beetle there. So when we try to incorporate the insect population dynamic into those system models, we're trying to looking at what's available in terms of the insect population models. So in general, we saw there's 2 types of models. First type of model looking at mainly focus on the insect part. For example, the individual-based modeling and the partial differential equation modeling that track the cohort, the density of a cohort. And then they all include the temperature dependence, and where it can be spatially implicit or explicit, depending on what type of model they have. And another type of model is called vegetation susceptibility model, like Landis model. Those models do not actually simulate the insect population at all, but trying to track the susceptibility of vegetation. So it's very easy to fit observations in those type of models, but because they ignore the insect population dynamics, they may not be able to correctly predict the future outbreaks, because those are nonlinear. So in order to build the model for the Earth system model, we want to both the insect part and vegetation part together and looking at the interaction at once. So what's the challenge to incorporate insect population models? The first one, clearly we see that there's different countries have different censuses, but there's no consistent insect outbreak data across different countries, and that could be a challenging component for model evaluation. And second part, in the past, we do not have appropriate vegetation models because insects may prefer bigger trees, for example, and have specific type of species, but most of those models do not differential size, and the plant functional type is just differential based on leaf phenology. So that's and the third challenge is that we do not have appropriate vegetation defense models yet. So there's different type of defense, like constitutive, like raisin production or biochemical defense, but it's not a lot of data on the model actually can help us to build a sophisticated vegetation defense models. And another component of the insect population model is that they normally account for the stochasticity of result from the environmental stochasticity or because individual variability because the genotype differences. But in Earth system models, normally we have a deterministic way. We do not want to incorporate the stochasticity because it's computation very expensive. And then another limitation is that the insect physiology, we may have a lot of data on mountain pine beetle, for example, but there's many different type of species, and we do not have a complete data set on different of those insect physiology. So we may only focus on 1 or 2 species at the very beginning. And finally, in terms of dispersal, and I think Matt's presentation clearly pointed out that dispersal play a very important role in terms of southern pine beetle to New Jersey. And but the data, it's very limited because it's very difficult to do those experiments. And also it's quite difficult to implement those dispersal in the Earth system model because Earth system model normally, especially on the land side, they are not good at doing the interaction at this point. very few models actually have the seed dispersal, for example. So how we can proceed ahead at the first step, look into incorporating the insect population dynamics. So I think this a great breakthrough in the vegetation part that can facilitate us to do the insect population modeling is that before we normally have big leaf model. We represent the forest as a single leaf layer and then do the photosynthesis and look in the carbon flux all together. But we do not represent the size and the canopy structure. But in the last decade or 10 to 15 years, there's a second generation vegetation model called a demographic vegetation model. They're trying to represent the succession dynamics, for example, forests of different ages, and then within each, the same succession stage, we represent different size of trees. So that actually enable us to better incorporate insect population, especially on the attack side. We can select big trees versus small trees. And this is the component mainly called COM-MED, and this has been sponsored by DOE, and Lano is actually involved to incorporate this ecosystem demography model into the COM model. So this provides us a great opportunity to explore new insect population and vegetation dynamics. And then second challenge in terms of stochastic representation. For example, this is a figure showing that the density of the physiological age for different time steps, and it's not just a single point, it's a distribution because of the individual level variability and environmental stochasticity. So Devin, as postdoc, come to our lab in this May, and I talked to him that we were not able to represent the stochasticity in the assessment model. And so what he actually have this genius idea is trying to capture this stochasticity based on a statistic theory-based convolution. Basically, he's trying to analytically derive the distribution of those physiological development for each time step. So in that case, we do not need to have the Monte Carlo type of simulation to capture stochasticity. Instead, we just have one single simulation, but we are able to analytically track the distribution change through time. And this is just showing for different mountain pine beetle development stage, looking at the dark color indicating his approach, just one analytic solution for the mean, and then the gray indicating multiple simulation of individual-based model, and clearly his approach is able to track the stochastic representation of an individual model, and that's great. And that's enables us to move to next step, looking at vegetation and insect interactions. So normally in the current insect population models, they do the vegetation defense in a very simple way. For example, if you want to kill a tree, how many beetle you need it, and that's it. But in reality, that may be much more complex. And at the Alamos National Lab, we have developed this mechanistic representation of carbon and water storage in the plant because we want to simulate the drought-induced mortality. But that's also provide opportunity for us to mechanistically represent the tree defense by, for example, the raisin production based on how much water they available in the plant and how much carbon available in their plant. So in this case, it is a potential opportunity for us to have a more mechanistic representation to represent the insect attack and vegetation defense altogether. And I'm really looking forward to that. And finally, when we build our model, it's important that we have the benchmarking data. We want to evaluate our model, how good are they? And clearly, we have this forest inventory data showing here. But another approach is that remote sensing. This is a paper by Arians and Jeff Hickey here, looking at Colorado using the Landsat and detecting the mountain pine beetle mortality. But this both approach advantages and disadvantages. For the forest inventory type of approach, we know what's the cause because you go in person. But normally very sparse, you only a few points in within a half-degree window. But for the remote sensing, we can do spatially extensive, but we may not know what caused the mortality. So in ideal case, we want to combine these two approaches together. And this is just a map by Matt Hansen from University of Maryland, he produced this global disturbances map based on the Lancet. And this is a big progress in this field, and this paper published in Science. And what we try, but one key disadvantage is that we don't know what's the cause of those mortality, and that will be difficult for model evaluation. So we have a student coming in this summer, and we're trying to using the remote, the FIA data, looking at the percentage of mortality based on FIA for the harvest, and then using the fire monitoring approach, looking at the percentage of the fire disturbances, and then using the drought threshold to detect what area are drought associated in a way. So in this case, we're able to attribute the mortality to different agencies. And if you look at the carbon loss, we can see that harvest caused most of the carbon loss, almost half of all carbon loss from 2000 to 2014. But drought or insect can be almost as twice as the fire. So it's a big component of the carbon loss, at least in US. And that's also a great justification for us to incorporate insect population and dynamics in the Earth system model. So finally, we're looking at the carbon consequence from the insect outbreak, but also may have great implication for the forest management. A couple of weeks ago, we just gave a presentation to DOD because they are concerned about how they manage their forest so that they can maintain their facilities on site. And traditionally, when we do the forest management in terms of fire, we just thin the small trees to suppress fire. But if you're looking at the mountain beetles, for example, this is in Black Hill, and those areas with harvesting in the past, you actually see the insects actually avoid them because they are too young. There's not enough phloem or nutrient available there. So the question is how we can manage our forest so that we can have the best carbon gains for local management, but also looking at the global carbon consequences altogether. I think that would be a very interesting discussion point. And what's the path forward in a way? So I've been in the community of tree physiology a lot of cases, but I do have background on entomology because I've postdoc at North Carolina State on that. But what I observed is that there's really a lot of interaction between those two communities, tree physiology and entomology community. They rarely talk. I will personally feel like it's great if there's more talk between those two communities and to coordinate their efforts together to make a better model for us to predict the insect outbreaks. And then another component I'm thinking that because the Earth system model, you normally look at the continental or global scale, then it's important that we coordinate different forest inventory data. For example, not only US, Canada, China, for example, and we have a consistent global data set on insect outbreaks. And another component I want to point out that as a DOE lab employees, right now, DOE Office of Science in the climate and environmental science, they pushed for the term called MODEX, model experiment integration. So when we develop a model, we always go to the literature and find the data we needed. But a lot of cases, you may not able to find the data you needed. So in that case, we may need new measurements. So it's a kind of model-driven experiment, and then the experiment can feed back to the model, and then in the end, we can help us better understand the whole system together. And we have both improved understanding and improved prediction by putting those mechanisms in the model. And another component I think I would like to touch on is that there's different communities in academia or national labs looking at different disturbance agencies, for example, fire, insects, or drought. They're different communities, but there's a huge amount of interaction between those different disturbance agencies. So it will be important for us to get together and looking at how different agencies, disturbance agencies interact with each other, because that's not a linear interaction, it's non-linear in the end. And finally, I want to point out that we need to identify the background mortality in our system model. So this is just a scheme showing that in the early age of the Earth system models, we have only one background mortality, and that's it. They do not respond to the future climate in a way. Recently, we have incorporated the second generation vegetation model. We have incorporated, for example, fire. We have incorporated the light limitation and some part of a drought based on the growth efficiency approach. But hopefully in the future, we can have more, for example, wind throw, insect and drought interaction, insect drought all together. So only a very small component will left as a background. Probably that's due to pathogen or diseases. So I think that's all for my presentation. I would like to acknowledge the funding resources from Alamos National Lab and also from Department of Energy, Office of Sciences. Thank you.
Thank you. Any questions that folks want to ask? Yeah, go ahead. Yeah, thanks for the presentation. Very interesting. So the demography model will help you with the stand age distribution, but how do you deal with the species composition within each plant functional type and how that varies with susceptibility to disease?
I think it will depend on the insects you're looking at. So we will select as, we will subdivide those PFT, for example, conifers into different species, different subgroups, so that's it's a host for a specific type of the insects in that case. So I'm not sure we find out the resolution like for all the insect species, but I think in the beginning we may able to define those PFT based on 2 or 3 major disturbance insects. Yes.
Yep. Go, Jim. This is Jim Stack.
Yes, I noticed in your graphic of potential causes of mortality, there were no pathogens.
Right.
And we are currently experiencing a couple of epidemics going through the U.S. causing substantial mortality. And in at least 2 cases, they're vectored by beetles.
Right.
So I was wondering, are you not accounting for that in your models? And if you are, I'm curious how that partitions.
I always like pessimistic, because we even have not incorporated the insect population model yet. So in terms of pathogen diseases, I feel like probably in the next 5 to 10 years, we may able to incorporate that. But I feel like at this stage, I think it's important to demonstrate that how we can incorporate insect population dynamics and to reasonably predict the outbreak and the insect-induced mortality. But I think I clearly agree with you. That's very important, and I think we should be on our agenda for the future.
If I could follow up, I guess I'm just a little concerned of your attribution to the vector in the absence of the attribution to the pathogen and oversubscribing the values of your parameters to just one agent.
Right. I think that's always a concern in the Earth system model in a way, but if you're looking at it in the past, most of those all attribute to the background mortality in a way. There's no response at all. So hopefully like with more, I agree with you, we will over attribute to a certain vector, for example, mountain pine beetles. But I think is that in the future we can have more pines there looking at the pathogen component, and if we have more accurate predictions in the future. But I think that this stage is very challenging. Yeah. Thank you.
Anyone? Keith's got a question.
Hi. So this is a question a little bit about the modeling philosophy, since I think you were describing your work here as all being targeted towards incorporating more features, more into the global Earth system modeling framework, correct? First part of the question would be, how much are you learning from an offline model approach where the insect model or other things could be run in a mode where you're taking output of the models and driving them and then not having the feedbacks in the model, or in a simplified, less complex framework? Because when I hear that the work is going into the Earth system model, that means there's this assumption both that the feedbacks are important to the system, that's why you're going to do it in the whole model, but also that they're going to be modeled well enough so that's adding value and not noise. So it's something about the sophistication of the level.
So I totally agree with you. When we add a new component of the models into the system, because it's already very complex, and the question is how much improvement you can make compared to the observation. And I think we need to test that using the data to see whether we improve the prediction of those tree mortality, for example, across U.S. Right now, there's even no evaluation of those models in terms of tree mortality across U.S., for example. We want to incorporate the insight to see whether improve that, and if we improve, then we will have the model run on both offline and coupled simulation looking at their climate feedback. My guess is that if you only consider the U.S., the feedback to global climate maybe not that much. So that's the reason I think really we need coordination across different countries to incorporate different type of insects all together. Thank you. Yes.
Yep, got a question from Ben Cook.
Yeah, so, kind of following on Keith's comment, how computationally efficient is ED? Because my understanding is that even in an offline capacity, running this ecosystem demography model, even over a limited area, takes a lot of time. And so if you really are looking to actually have this incorporated into Earth system models, there's a concern about, whether that computational trade-off is worth it, adding it into these models, if it's going to be a big drag computationally.
Yeah. So that's a very good question. As far as I told, most of the assessment models, I think, or CSM, the COM components only consume 1/5 of the total the assessment model simulation. So it's a small proportion. And always get the message that even you get much more complex, it should not be an issue because already consume less. But from a practical perspective, we do see this computational cost increase.
Have you run the ecosystem demography model?
Yes.
So
yes.
how long does it take over?
It depends on the threshold, how many cohorts you want to put in. You can run for each grid cell 100 cohorts or like 10 cohorts in a way. So it varies, but I think it's not like double. It did not double at all at this point. Yeah.
Yes, Barbara?
So I was just wondering what the temporal scale, not spatial, but the temporal scale of the climate information you...
Yeah, so in the Earth system model, normally we run a half an hour simulation.
So the climate data coming in, driving what's going on is on a half an hour?
Right.
Okay.
So I'm just going to ask for folks online that are watching, if you have questions, this is your opportunity. And anyone else in the room at this point? So I thought it was interesting where you mentioned you talked about your solutions and forest management, and you showed that picture of whether we're thinning out the small trees versus taking out the big trees, and any thought to, I guess I hate to say this, but removing everything is really a solution we don't want to go towards. No, and so the thought of from your group and maybe others in this room about avoiding which direction to go, it's really an interesting kind of scenario.
Yeah, I think I do not have a perfect answer to that, but I think like Jeff or Bob or Matt can add on. I'm thinking that depends on your location. You do see what's the dominant force of disturbances. If it's fire or insect, then you may manage different way.
And that's a good comment, because I think what I've heard in a lot of the talks today is this kind of microsite variation, which applying models is important, and I understand that, but there's variation at these small scales that also has to be accounted for. So if there's nothing online and we're not getting any questions. Okay, so there's a question from a previous talk. So thank you again, Chonggang. Yep. So we have a question online for
Matt Ayers.
For Matt Ayers.
And I will read it verbatim. This is from Susan Kaler. Aside from temperature effects, has there been any data analysis in terms of population trends on the movement of products from infested area in the south to New Jersey that may harbor the SPB pest?
Thanks for the question. Yeah, that possibility has come up. There don't seem to be any obvious vectors or pathways for human transport of southern pine beetle into New Jersey or from New Jersey to Long Island. Bark beetles aren't very well suited to being moved on wood material because they leave trees very early. And trees are typically not moved after that time. So it's not very far from places like Virginia and Delaware where southern pine beetles were before, or from New Jersey to Long Island. So it doesn't seem problematic to imagine that they just got there themselves, and they probably have before. It's just that previously they used to get whacked back by a cold winter before anyone would notice.
Okay, thank you. So there's no other online questions. Anyone else have any questions or want to follow up with something they heard earlier? We have about 10 minutes, and so I'll just, if there is something that someone wants to bring up, I'd say, now would be a good time. Yes, Liz.
Hi, I was, struck, Matt, by your comment about the invasion fronts moving back and forward, and I wondered, if there has been a historic link to pine bark beetles in the north, if anyone's done a genomics analysis to see, sort of what the historic relationships have been, to see if that's a path of understanding if the beetles that you're seeing today have had a history there in the past.
Yes, I understand your question. There was some work that was done on microsatellites of southern pine beetle, which is now 5 or 10 years old, and as you know, that technology is improving a lot. So I have some colleagues who are trying to do some of that work right now, and I think it's going to be very exciting to see what they come up with.
Okay, thanks.
So I might add about some work we've been doing in the West with mountain pine beetle. There's definitely a signature of selection molecularly showing that those populations that are in Canada basically are new. So they after glaciation, the pines started going back in and the insects have then followed. Some recent work we've been doing, similar to what's been shown with tree species, is that the glaciation also resulted in patterns in the western US as well. And in fact, for mountain pine beetle, it's pretty interesting. It was separated. There was barriers, which we now see in not only just in the genetic markers, but also when you take them into the lab. Like you take an insect, mountain pine beetle from one side of the Great Basin and one from the other side of the Great Basin, and you try to mate them in the lab, the offspring are sterile. So that reproductive isolation has been there long enough for speciation to be starting. So and the patterns that we see follow directly, which makes sense, the patterns that have also been shown in a number of pine species, which is their host. So.
Okay, good. Thank you for sharing that, Barbara. Anyone else? So I guess we're going to adjourn for today. I know it's a little early, but that's okay. So we will reconvene tomorrow morning. If you look at your agenda for those folks online, we will be starting at 8:30. And so we will go ahead and end for today. Thank you again for the speakers presenting. Great information, great start for the discussion that I anticipate will continue, and we'll look forward to hearing more tomorrow. So we'll see you then, and thanks, everyone.